So this one is physiology of protein digestion, absorption, and metabolism. So under digestion, if you can write this point like this, you get your full mark. Then under proteins, digestion of proteins at stomach. What happens in the stomach? Pepsin, there's this enzyme equal to pepsin. Aya activates a hydrochloric acid get stomach. Then you break down proteins into what? Peptides. Remember, in the stomach digestion, protein is breaking down into peptides. That's number one. Then in the small intestine, what happens in the small intestine? Get amadia under proteins when you eat proteinous food. Digestion of proteins in the small intestine. Pancreatic enzymes. What are the pancreatic enzymes? We have the trypsin, chymotrypsin. These are the two pancreatic enzymes in the small intestine. Further, I break down peptide. What's peptides? Peptide are the storage form of hydrochloric acid. They break down proteins into peptides. Though these peptides are the protein molecules. Break down into smaller peptides. That's the enzyme, pancreatic enzyme. I break down peptides into smaller peptides. Then there's this other brush border peptidases. There's another enzyme called brush border peptidases. Converts peptide into amino acid. So amino acid now is the storage form of proteins. That's under digestion. What happened? Protein digestion in the take place in the stomach and small intestine. And the storage form of protein is what? Amino acid. Then under absorption of protein. Then this amino acid that was broken down from protein, amino acids are absorbed. What is absorption? Are distributed into the bloodstream through active transport. What is active transport? Active transport only require energy. Active transport, I use ATP and NADH. So I use active transport. I distribute into the blood capillaries, bloodstream in the capillaries. I absorb all these nutrients, these amino acids. Amino acids, the protein molecules, in the storage form of proteins into the intestine villi. Intestine, there's a cell in the intestine villi. That's absorption. That's the part that absorbs amino acid. Then under metabolism of proteins. The storage form of protein, there's the number 1 point. Amino acids are used for protein synthesis. And synthesis that they use building. I mean, any place you see the synthesis that they use building, and no place they're breaking down. So the amino acids they are used for protein synthesis. Then structural, there's what we call structural and function proteins. Are built up from amino acid. Then another point under metabolism, the excess amino acid undergo deamination. That copper mentioned for the class. What's the meaning of deamination? Deamination is the process by which the amino group, let's say protein, the storage form of protein, is removed from an amino acid. Deamination, like when you have an amino acid here, then you remove the amino acid group. So it's the process by which amino group is moved from an amino acid, producing ammonia. So deamination is a process by which amino group, as NH2, the amino group, is removed from amino acid. Then this process that produces what? Ammonia or ammonium and keto acid. So this process is essential for protein metabolism, yes. So deamination is essential for protein metabolism. Then what is deamination? In case of a objective, is a process by which amino group is removed from amino acid, producing what? Ammonia or keto acid. That's deamination. So deamination in the liver, this deamination happens in the liver, producing ammonia converted to urea and keto acid. So the process of removing amino group from the amino acid is deamination, and it's taking place in the liver. And this process produces ammonia and keto acid from ketone. Then this keto acid can enter Krebs cycle. From keto acid, they enter Krebs cycle to form what? For energy production or gluconeogenesis. So that takes us to gluconeogenesis. In case of whether I will define gluconeogenesis, may define glycolysis and glycogenesis. So this one I did say gluconeogenesis. In case of get objective, but what is gluconeogenesis? So gluconeogenesis is the process of synthesizing. What did I say is the meaning of synthesizing? Building up. It's a process of building up glucose from non-carbohydrate source. What are the non-carbohydrate sources that we have? Fat is all carbohydrate, protein is all carbohydrate. So gluconeogenesis. When you heard of Genesis said, Genesis Genesis in the Bible now. New beginning of new glucose, building up of process of building up of glucose from new glucose from non-carbohydrate sources such as amino acid, glycerol, and lactate to maintain normal body glucose level. So that's gluconeogenesis.